Uh, Let me begin by thanking the previous presenters for their excellent presentations. Gerard Liebisch, Michael Holopek, and Maria Fedora have very nicely described the detailed methods, saving me any details about the methodologies I'm going to discuss. And Bob Murphy, especially, for having introduced and insisted on absolute quantitation as Lipidomics developed its lipidomics. And he very nicely explained the importance of internal standards and isotope dilution. So I will just be able to refer to that. So I have been asked to speak today about quantitation of free and esterified fatty acids and eicosanoids. And I've got to advance here. Okay, and I'm going to do so in two parts. First, the shorter part, I'm going to focus on fatty acids. And for that, we use GC mass spec, which I will briefly review. And I'm going to do two parts, free fatty acids and then esterified fatty acids. Then the larger part of my presentation is on lipidomics of eicosanoids and related oxylipids, for which we use LC mass spec. Now, let me pause right here and make clear. The word eicosanoid technically refers to 20 carbons, icosanoic acid, for example, for arachidonic acid. However, it has grown up to be used loosely to mean oxygenated polyunsaturated fatty acids. And I always referred to eicosanoids and related oxylipids, even if I only say eicosanoids. And that includes fatty acids generally from 18 carbons to 22 carbons. Now, I'm going to again talk about free eicosanoids and then esterified eicosanoids. And those are using triple quad. And then in the last brief part, I'm going to talk about directed untargeted approaches to eicosanoids, which take advantage of a hybrid orbit trap and exact mass capability. So for the students present, I want to begin by kind of summarizing the approach that Lipidomics took to the field of lipidomics, which we called the CLASS approach. That stands for Comprehensive Lipidomics Analysis Using Separation Simplification, a so-called divide and conquer strategy whereby the specific protocol and methods depends on the category of lipids one is analyzing. And all the previous speakers today really spoke to details of how one does it for a particular category or one or more categories of lipids. So just as a brief orientation and summary of that, let me say one starts with whatever biological sample, cells, tissues, plasma, doesn't matter. In some way, one has to get that biological sample into a homogenate, which would be an aqueous solution in general, but a homogenate so you can sample it in a quantitative manner. The first thing you do is add internal standards. And these internal standards are category specific depending on what lipids you're targeting. They can be deuterated, odd chain, or other variables. Then one carries out an extraction. And the extraction is critical because you want to extract all of the lipids of the category you are looking at. And there are liquid-liquid extractions, SP, and others. And previous speakers this morning or this afternoon for some of you, or evening for those in Asia, have many of the details. Once you've extracted it, then you need to analyze it. And to the degree that the field has moved to separation technology from the old days of thin layer chromatography, first to GC, and now, as you can see, abundantly to many forms of liquid chromatography. And for each category, the solid support differs, as does the solvents and the solvent gradient. And then one comes to the mass spectrometer variables, and you have different types of mass spectrometers. You have additives, ionization mode differences, and mass spec monitoring modes. So each of these are designed for what category of lipids or categories one's looking at. So this is the GC mass spec system that we use and have used for a long time in our lab for analyzing fatty acids. Now, the disadvantage of GC, as was noted by previous speakers today, is that you must have your analyte in a form that it can pass through the column in the gas phase. And fatty acids with a carboxyl group cannot. So one has to derivatize fatty acids. Initially, people used methanolysis to make methyl esters, and subsequently many specific derivatization agents have been developed. We happen to use for our fatty acids pentafluorobenzyl bromide derivatization, and then it's very efficient in dissociative electron capture ionization, so we get very clean results with that. Now, just to start off and emphasize, I think what Bob Murphy said was, the first thing one does is look at those internal standards. And this just shows you pure arachidonic acid, a primary standard, weighed, pure and weighed, and then put in solution very quantitatively, including some D8 arachidonic acid. They come with the identical retention time on GC for the derivative, but the molecular weight is different. One is 303 and one is 8 units higher at 311. And we use 14 deuterated internal standards for our analysis. This just shows you the deuterated internal standards are nicely spread out in a 15-minute GC mass spec run, and those give us our quantification. This is just a typical sample from mouse liver looking at the free fatty acids and quantified against the internal deuterated standards. What's shown here is that the majority of the free fatty acids are palmitic acid 16O, oleic acid 18.1, and stearic acid 18O. That is the two major saturated fatty acids and the major monounsaturated fatty acid. But one also sees a good peak of arachidonic acid and DHA 22.6. So to summarize what I've presented about fatty acid analysis, we use in our lab the following protocol. The first thing we do every time we're going to run the GC mass spec is do a standard curve. We have a mix of 37 primary standards. Again, primary standards are pure, weighed, and then put into solution, into a solvent solution that is very quantitative, and we include a mix of 14 internal deuterated standards. We make that mix, we remove the solvent, we derivatize it, we take up the derivatized material in isooctane, run the GC mass spec. Now, we do standard curves for by isotope dilution to standardize those primary standards against the isotopes. Now, if we're looking at free fatty acids, we take our sample, we immediately, if one, as you would say, takes it out of the -80 freezer, aliquots it, and immediately adds the internal standard, brings it to volume, we carry out an isooctane extraction of that. We remove this, then we remove the solvent derivatives and do the same procedure. Now, if we want to know the total fatty acids in a biological preparation, that is all the fatty acids that are esterified, at least 2 triglycerides and phospholipids, which is what we tend to focus on, we have to extract the biological sample first using an extraction as has been described earlier today by others for all of the categories one wants to see. And then we take that material and we subject it to alkaline hydrolysis under very controlled conditions at 37 degrees for 30 minutes. Then we quench it by lowering the pH and do an isooctane extraction, remove the solvent derivatization, and run the GC mass spec. And this is the standard protocol for alkaline hydrolysis. Now let me turn to the more elaborate procedure that I want to describe today, and that is the eicosanoids and related oxylipids. So let me start with the instrumentation. We use a Waters Acuity UPLC system linked to a SciEx 6500Q trap triple quad for this analysis and reverse phase. Now, the first difficulty I want to point out about eicosanoids is that there are many isomers in them. Here are 3 of the major eicosanoids, and PGD2, PGE2, and 11-beta PGE2. These are isomers of one another. Their exact mass is exactly the same. They fragment identically, as illustrated for PGD2 in the second quad of the triple quad. So we rely on UPLC to separate these 3 isomers by retention time, and then we analyze them by MRMs using the same MRMs and the same conditions for each of those 3, but we've separated them by retention time, showing the power of UPLC. Now, in our standard eicosanoid analysis, we look at some 200 eicosanoids and related oxylipids in a 5-minute UPLC run that we have developed. About half of the eicosanoids are derived from arachidonic acid, as shown symbolically here. The other half are derived from fatty acids of 18 carbons, 20 carbons, as well as 22 carbons like DHA. And we include 26 internal deuterated standards in the mix. So that tells you that each internal standard is used for 5 to 8 primary standards of very similar composition and similar fragmentation, so that the MRMs that we compare for the primary standards or the deuterated standards where it's not identical can be relatively the same, as Bob Murphy described the methodology earlier today. So the protocol is we do a sample extraction, we run it on LC-MS. Basically, it's a 5-minute run. We can identify by MRMs, and because these are all targeted where we have primary standards, or in a few cases, very close relatives where they're not available, but we know enough about them to identify the fragment. We can do variables like time courses of a cell culture or plasmas of different individuals, and we can plot them out in heat maps and look at other fancier plots to compare the peaks and do pathway analysis, as all of this will be discussed in some later sessions in this meeting. Okay, I want to now turn to alkaline hydrolysis for total icosanoids, because this brings in some complications. So what is shown here is now we are actually doing lipidomics of phospholipids and lysophospholipids. I'm not going to talk about how we do that, because others have, but shown on the left at the top is PC, second is PE, and third down is PI, with no base hydrolysis. There's the peak for this phospholipid in a mixture. And then what happens at minimal hydrolysis conditions that I showed you for the fatty acids, 37 degrees for 30 minutes, and then at some more stringent conditions. All the PC is gone, all the PE is gone, and all the PI is gone. So all the phospholipids are completely hydrolyzed under these conditions. What remains is SM, that's sphingomyelin, because these conditions don't hydrolyze sphingomyelin fatty acids on the amide. Also, ethers or plasmalogens are resistant to this hydrolysis condition, but we're hydrolyzing all the free fatty acids from those. And on the right is shown the lysophospholipids that also are completely hydrolyzed, the remaining fatty acid on it under these conditions. So this looks good. We can release the free fatty acid from the esterified. However, there's one little glitch with it, and that is that some eicosanoids are destroyed by base conditions. And I've just picked one here. This is a oxidized derivative of DHA, and it shows no saponification in increasing conditions, and it's plotted on the bottom, that as you increase the stringency of base hydrolysis, you see a decrease in the recovery of this primary standards. And if we look at it in more detail, and I just want to start by looking here at free oxylipin standards in their free form. When we look at free oxylipins against our internal deuterated standards, we have some minor losses in extraction for some of them that are normalized by the internal deuterated standard that we use and have calibrated and our standard curves. So thromboxane, PGE2, PGD2 we get 100% quantification by normalization with the internal deuterated standards for losses in extraction and other procedural aspects. In contrast, this is for free form without alkaline hydrolysis. When we do alkaline hydrolysis on complex lipids, and we look at alkaline hydrolysis conditions with standards of PGE2 and PGD2, we get no recovery. Why? Because they're destroyed in the presence of base. In contrast, thromboxane B2, 5-heat, and others have some minor losses, maybe extraction, maybe base, but using normalization with the internal standards, we can calculate and quantitate 100%. Why is there losses of some eicosanoids? Because there are a number of breakdown mechanisms described in the literature: dehydration, oxidation, base catalyzed rearrangements of some of these molecules, and that happens more frequently at higher pH and base catalyzed conditions. So in summary, when we look at these couple of hundred eicosanoid standards in our standard runs, when we're looking at them for presence in total lipids, we are limited to about 50 of these that we can actually reliably measure. And they're illustrated here, for example, of the 50 COX derivatives that we usually look at, only 15 are reliably recoverable with base hydrolysis. Shown in red are some of the structures that are destroyed by base. In green are structures that are recoverable and measurable. So using this method, let me now summarize what it is saying, and that is that when we're doing icosanoids and related species, the very first thing we do is we take 147 primary standards in our mix. These are, again, pure pure samples, and weighed, measured, diluted, and use isotope dilution to do standard curves, and 26 internal deuterated standards. Make the mix, remove the solvent, take it up in buffer, and do our UPLC mass spec approach. When we're looking at free oxylipins, we take the sample, we put our internal standard mix in, we bring it to 1 milliliter of water. We do our extraction, which in this case is SPE, and we then remove the solvent and run our UPLC. By the way, we take up our solvent in our sample in a milliliter of aqueous solution, but it contains 10% ethanol, and we use the trick Bob Murphy referred to of precipitating the proteins out to get them in, the icosanoids into solution, and that works. Now, when we're doing total oxylipins, we do the same scheme as we did for fatty acids. We do include BHT in the mix to prevent oxidation. We're very careful about the temperature and time. We do our quick quench, we do our SPE, remove the solvent, and do our analysis and look at the results. And when we do this in human plasma, and this is based on looking at human plasma with standards present, and when we're looking at free icosanoids in human plasma versus esterified, for those which we can reliably measure, we find that somewhere between 20 and 80% of each of these icosanoids is present in the esterified form or as a complex lipid, mostly probably phospholipids, but also triglycerides. And so the majority of icosanoids are not free. They are in complex form in human plasma. In stimulated macrophages of the sort that I discussed yesterday, where we stimulated with LPS and ATP and looked at icosanoid stimulated production, and then looked at esterified versus free, we see that there's a broader range. These particular icosanoids tend to range from 0 esterified to 100%, depending on the particular icosanoid. And so this is just an illustration of the kind of data one gets. Okay, in the last few minutes, I want to turn to one last subtopic, and that is that, yes, besides the triple quad, we also look at icosanoids with exact mass using an Orbitrap, and we actually use the Thermo Q Exactive hybrid Orbitrap setup that's shown here with reverse phase. I'm going to just show a few slides briefly about icosanoids. While this is up, I should mention that while originally we did phospholipids and sphingolipids and complex lipids on the triple quad, we have moved that technology to this machine now. And Jürgen Hartler, who will give the last talk tomorrow, Wednesday, will describe his new developments of the LDA system for identifying and quantifying sphingolipids. We use that system for phospholipids and sphingolipids on this instrumentation. but I'm not going to speak about the phospholipids as such. Now, to introduce what I'm talking about, we've all
I'm
been
sorry, Ed,
talking-
just tell you that we are running a bit out of time, so.
Okay, I will finish up in just a few minutes. We're talking about on the right targeted, and this is the same process as has been described. I want to contrast that with what we have termed directed non-targeted analysis. And it's summarized on really this single slide. If one looks in the Lipid Maps database, there's some 1,100 putative oxylipids, but we only there are only 225 oxylipid standards commercially available. And if you look at these oxylipids and just look at their mass to charge in the upper left, there's an enormous number of isomers at each m/z, even with exact mass. If you look at the extracted ion chromatogram over the retention time separated, one gets a nice separation of all of these standards. Now, taking advantage of the chemical knowledge and the mass defect that earlier speakers have talked about, if you're at a very exact mass between the number of carbons and those compounds with more oxygens like icosanoids, which tend to have a high oxygen to carbon ratio, have a different mass defect, and separate by mass defect, we can identify and spread apart on an exact mass basis which oxylipins are sterols, endocannabinoids, phosphors, or bile acids, and therefore identify by color coding those that are likely icosanoids. And using that approach, when we took large numbers of plasma samples, some 1,500, stratified by age or BMI or C-reactive protein, and ordered them by mass to charge ratio, while some number of those peaks are known standards that we always target, some are putative known compounds by the calculation method in lipid maps. So they're sort of identified but not shown to be. And many peaks are novel because they don't fit any of these criteria. So this is a methodology for trying to identify new and novel lipids by a directed, non-targeted approach. So let me conclude by thanking my collaborators and co-workers. Just mentioned those in yellow: Ozzy Queenberger, Aaron Armando, and Signa Dahlberg-Wright, who contributed to some of the data shown here. And the last part was in collaboration with Jeremy Watrous and Mo Jain. And let me now open it for questions and mention again that I have a funded postdoctoral position available to follow up on some of these approaches with cellular lipidomics, eicosanoids, phospholipids, and sphingolipids, and email me if you have an interest in the description. Thank you.